Okay, we are going to be going over DNA replication. I'm just going to write over here, we have our lagging strand. So we're going to go over the two different strands that come out of DNA replication. We're starting over here and moving over here. We have two strands, the lagging strand. And let's just do orange. We have your leading strand. That's down here. Okay, so again, starting over here with one DNA strand, your topoisomerase is going to help unzip it. So your DNA is becoming unzipped. And your helicase is going to break the hydrogen bonds between these corresponding bases right here. It's going to break the hydrogen bond. And as you can see, they're being separated. So you're losing your hydrogen. The bases, the corresponding bases are broken apart, and the strands move on. Now, your DNA polymerase. Let's go to the leading strand. Your DNA polymerase is going to take these bases from the original strand. It's going to take DNA primer or RNA primer from the. It's going to take RNA primer and it's going to bind it to the original strand. And it's also going to act as a signal. It's going to say, okay, we start binding here, we stop binding here. It's going to follow along with the DNA strand. So here we have this is like a checkpoint of sorts. It's like just letting you know what's happening, and it's also just helping assist binding these corresponding bases together. So your RNA primer here is being bound to the DNA with its corresponding base, so AT and GC. Uh, and then over here at the top, so hold on, because the RNA primer has no gaps, we know that it's continuous. It's a continuous cycle. Now, if we go to lagging, you can see there's gaps here in the RNA primer. So it's getting to the DNA primase. Uh, and then you have the RNA primer. And you can see these gaps. These are called the Okazaki fragments. And this is why it's a discontinuous cycle. It's a discontinuous cycle. You have the corresponding bases being attached, and the DNA polymerase is going to attach them. It's going to attach it again. It's going to read it. And it's going to be like, okay, we need to start and stop here. That marks the start and stop of the strand of DNA. You have your corresponding bases attached, the hydrogen bonds are recreated, nucleotides repaired. You have all this done, but because there are these gaps, they need to be fixed later on, making it a discontinuous process. The fact that this doesn't have the gaps in the RNA primer prevents it from being discontinuous. Um, Now that you have two strands of DNA, your DNA ligase is going to seal up the sequence of DNA into two continuous double strands. Then your two strands of replicated DNA are created, and your body is going to proofread the DNA. It's going to make sure that there are no errors, and it's going to do its best to uh, avoid mutations, any kind of insertion, deletion, any kind of thing that's going to lead to a negative impact on the body. And that is DNA replication.